project. So the DNR is funding it and leading it, doing all the data collection and the project management and a lot of the analysis, but it's a huge project and it's extremely complex. So we have a lot of partnerships as well. We've got a couple scientists at the USGS National Wildlife Health Center down in Madison, and the University of Wisconsin-Madison as well. So if I can forward a slide here. There we go. I never know to what degree people are familiar with chronic wasting disease, but so I'll just kind of give a brief rundown here. So it's a very strange disease. It's caused by misfolded proteins. So it's not a bacteria or a virus or anything like that. There are these misfolded proteins, and when they come into contact with the normal variant of the protein, they can cause the ones they come in contact to misfold as well. And they don't degrade or kind of cycle normally within the body. They aggregate. So they continue to build, aggregate in the brain, and eventually that will disrupt the neurological function. The disease is, so again, this is in a class of diseases sort of like Creutzfeldt-Jakob, that's in humans, mad cow, which you may have heard of. It's different from mad cow in that this is infectious. Mad cow wasn't infectious. It's hopefully, knock on wood, different in that humans don't appear to be susceptible, although that's ultimately an open question still. So this disease is always fatal in deer, and so that's really important, obviously, but also important is that it has a long disease course. So they don't die immediately. It's not like rabies where they get it and die soon thereafter or recover. They get it, they'll never recover, but it can take a long time. That has a lot of implications for the potential impact on the population. As initially, there's no at least outward changes once the infection starts, outward changes to the deer. Eventually, what will happen is they'll become emaciated, and at the very end they'll lose coordination and awareness. We'll often call them like droopy droolers. So you'll see them, their head will be down, their ears will be down, their tongue will be out, and they'll just sort of be trembling and unresponsive, and eventually they'll just tip over and die. So this picture on the right is one of our collared deer that had chronic wasting disease and was the end-stage chronic wasting disease mortality. And it's kind of hard to see with this picture with its winter coat coming on, but if you know what you're looking for, if you look close, you can see that the spine is really prominent. You can see the shoulder bone really prominent, and that rump muscle is extremely atrophied. So this deer was extremely emaciated, so it has lost entirely all its fat reserves, and not only that, but it had lost a significant amount of muscle as well. That's really common. This is just basically the common way that it ends for them if nothing else catches up with them first. There's no immunity that we know of. There is some genetic variability and susceptibility in the disease course, and what I mean by that is when you do sort of population level studies and you look at the prevalence of the disease across different genotypes, there is a genotype in which the prevalence is lower, but it's not zero. So it's not as though that genotype confers immunity. It just means they're less likely to acquire it. And then it appears that if once they do acquire it, that it does take even longer for them to succumb to the disease. This could be a whole talk in and of itself, but I think from what we're starting to learn here, this genetic variability, susceptibility is probably not going to be relevant for management or have any relevance for, while we are all alive. It's just that strong of a selective force, apparently. So the disease is shed from infected deer through saliva, urine, and feces into the environment, and also, of course, infected deer that have the disease that rot in the landscape. That area, of course, will have prions in it. Transmission can be direct, meaning from individual to individual. Deer groom each other a lot, so they're licking each other and stuff, and so that's one possible way for them to get the disease. But then also like what we call indirect or environmental contact. So again, when either through feces, saliva, urine, or whatever, that the prion remains infective in the environment for some unknown period of time, and deer can uptake the prions that way. That's been demonstrated, for instance, in pen situations where a bunch of naive deer, uninfected deer, are put into a pen that is free of other deer, but had previously had infected deer, and then those deer became sick, so they got it from their infected environment. So we know it can happen either way. We don't know the relative importance of those different transmission mechanisms, which is a huge unknown and really makes things difficult to, for instance, forecast the future dynamics. This is a current North American picture of the disease. Every time, so if you want to see the picture of the disease, you can just Google CWD USGS CWD map, and you're going to go to the USGS National Wildlife Health Center web page that keeps a current map, and I cringe every time I click on it because it's always getting worse and worse and worse. You can see the dark gray is kind of the sort of original known distribution that's in Colorado and Wyoming, but it's spread far and wide since then. And of course, this is our known distribution. Obviously, there has to be some sort of surveillance occurring to be aware of the distribution. So it's certainly the case that there are places not represented on this map that have infected deer that we're just not aware of. That's always important to keep in mind. This is a current snapshot in Wisconsin. And the same kind of thing applies, just because we haven't found it doesn't mean it's not there. But on the flip side, I hear at least people say, well, it's everywhere. If you look for it, you're going to find it. And that's not true. Our surveillance is not perfect everywhere, and there's certainly infected pockets of deer that we are not aware of. I'm sure that's the case, but we have done really good surveillance in most of the state, and we at least can say that it's not just evenly distributed. It's not just everywhere. And there's where we have really good surveillance in the southwest part of the state, where we have our prevalence, we see really strong spatial gradients in the disease. So it's not just everywhere. It's a true epidemic. It's currently in, I want to say, 26 of 72 counties, something like that. Okay, and so just as an example of, I just showed you more or less the current distribution that we first discovered it in Wisconsin in 2002, and so the distribution, geographic distribution in the state has increased a lot since first discovered it. And prevalence in those areas, especially where it's been around for a long time, has also increased. So this is just one example. So this is a time series of chronic wasting disease prevalence in one part of Iowa County. Iowa County is west of Madison. That's where that's sort of our epicenter. This is one of the higher prevalence areas, and this shows for adult males. So that means for us, a 2.5-year-old or older. This comes from hunter harvest surveillance. So hunter harvested deer get tested for chronic wasting disease. So we see that, when it was first discovered, prevalence was quite low, and it stayed low for a number of years, and then it increased quite dramatically, and now we get to the point where it's nearly a coin's flip. When you harvest an adult buck, it's almost a coin's flip whether or not it's infected. We don't know how long the disease has been in Wisconsin. We only know when we detected it. It could have been, decades or a decade or more prior to detection. We don't know, and we'll probably never know. So it's a fatal disease. So we're worried about, naturally, we're going to be worried about the effect of the disease on survival in deer populations, but it's also a chronic disease. So that which sort of, attenuates that potentially. There have been other studies, especially out west, in fact, almost exclusively out west, about looking at the impact of chronic wasting disease on survival. And what you're seeing here is a survival curve plot that compares uninfected versus CWD infected deer. These are mule deer in Colorado. They see, indeed, like you'd basically expect, that there is an impact on survival. we wouldn't want to just assume that what they found in Colorado and an entirely different species, although related, is going to apply to Wisconsin. Our deer have a different set of risks than their deer do. So we want to find out what this is for our deer.
Hey Dan, can I interrupt and ask a question for a moment?
Yeah, sure.
And I want to do that for two reasons. First, to ask a question, but second of all, to demonstrate to people that you're welcome to raise questions during a talk. You showed us the hunter surveillance plot, and then you're showing us this, the increased mortality in infected animals. Do you think hunters selectively are more likely to get infected animals because they're sort of have less mental acuity, or they're just easier to harvest for the hunters?
It's possible. I'm trying to think. That will be something that we ought to be able to learn with this project I'm talking about. I'm trying to think what, and I should know off the top of my head, but I'm trying to think what other studies have found, and I can't recall. I don't. Every different area has different intensities of hunting, and especially out west, you've got mule deer that are already not as, they're in a more open environment. They're already not as wary as white-tailed deer. So there's all these differences, and so in terms of what might be happening with us, it's certainly possible. We should know, but we don't. I guess we don't know at the moment. So yeah. So sorry. I should. Yeah. I'm just trying to think of the literature on that, and there hasn't been a lot of that, but it has been studied. But I don't quite recall, but we should be able to find out.
And then I have another question that somebody chatted in. I thought CWD, chronic wasting disease, was unique in prion diseases, that the prion was also in the skeletal muscle. So does it differ from, I think in human prion diseases, it's in the brain and maybe the spinal fluid, but do you know?
Yeah. So yes, it is found in skin and muscle. We're actually collaborating on with some researchers at UW-Madison who are the prion sort of experts. We're giving them skin samples for them to be able to see if they can detect it in skin. And a lot of it has to do with your detection methods and the sensitivity of the methods, but it's not, in terms of like where it's found within the body, it is not restricted to just the brain and the spine. Of course, there's nerves throughout the body. So when you say it's like in the muscle, I don't know. I honestly don't know. I mean, it might be within the body, but regardless, it's definitely not restricted to the brain. And so, for instance, it's in the lymph system, because that's what we often use to test lymph nodes and lymph follicles. So it's
I think Uwe Pot has a question here, I believe.
Okay.
I don't know, can you chat that, Uwe?
Yeah, I have just a quick comment. I lived in Switzerland at the height of the mad cow epidemic over there, and of course, thousands of cattle were slaughtered at the time. So people did not want to take the risk to eat muscle meat even, and I think the point really is that nervous system, nervous tissue is everywhere, right? Also in the muscles, really. So yes, I mean it was apparently also present in cows in those tissues.
Right. Mm-hmm. So, I'm making the point here that we have good reason to think that chronic wasting disease is going to be a factor that can impact deer populations, but it's always going to be one, if anything, it's going to be one of other factors. And hunter harvest is, when present, is usually the dominating factor, in at least in terms of, in Wisconsin, especially in Midwestern states, is hunter harvest is by far the fact that influences populations the most. And so, we're interested in CWD impacts. We have to put this in a greater context in terms of what are the other drivers of population change. And so this plot is just a time series of recent hunter harvest in Iowa County, which again is that county that contains most of the epicenter, and with CWD. And what we see here, this is due to big regulation changes, legislation and such, which we won't need to go into, but we see our antlerless harvest has declined dramatically. So that's just another thing that we need to make sure we're considering. So, for instance, we know that CWD is prevalent in Wisconsin, so you often get the question of, well, why there's still a lot of deer there? Why is that? If it really was a population factor, wouldn't it be obvious? Why wouldn't you just need to do a study? Well, because there's other stuff going on at the same time. So at the time where we presumably, possibly had increasing impacts of the disease, we've had decreasing harvests, which obviously counteract. And then the other thing is, another thing is recruitment. So recruitment, for what we consider recruitment in wildlife, is it's kind of defined as the number of young that enter the fall population per adult female. And so that's a function of pregnancy rates and litter size, and then fawn survival through the summer. And so we have, for ages and ages, done, the DNR has done late summer, early fall surveys of deer recruitment. And in that region, the southwest portion of Wisconsin, we've seen a steady decline in recruitment. And so this is important because this is, how many more deer are you pumping into the population? Obviously, that plays a role in how many you can take out of the population every year. So the more productive your population, the higher the mortality, adult mortality, can be sustained. So whatever our ability of the current population is to sustain chronic wasting disease and hunting it, it isn't what it used to be. So in the 80s, the survey of fawn pregnancy rates, so about half of the fawns, female fawns, were pregnant. And what I mean by that, fawn pregnancy rates, that means deer getting pregnant during the first breeding season of their life and then giving birth about on their first birthday. That's what fawn pregnancy rates means. So in the 80s, again, it was about 50%. That's a really highly productive white-tailed deer population. That's a deer population that is far below the capacity of the habitat to sustain it. There's lots of food and resources for all the animals, and therefore they were just really cranking them out. Well, that's changed quite a bit. The deer population is increased quite a bit since then. And when we did a recent survey a few years ago, pregnancy rates have almost halved. So it's my theory is that it's based on this higher deer density and reduction in the availability of food in the landscape and reduced nutrition. That's my theory. There's also the potential for a role of predation. For instance, bobcats, they're common now in that part of the state. They weren't. In fact, they may have been extirpated decades ago. They certainly weren't common. Coyotes, we don't have good data on this, but coyotes are possibly, likely more abundant than they used to be. Certainly, when you talk to the locals, they say that there's a lot more coyotes than there used to be. So there's a potential role of predation there, and because these predators, coyotes and bobcats, mainly prey on very young deer. So again, the bigger point is, when we're trying to understand chronic wasting disease, we need to put things in a greater context. So what is the future of deer populations? That's the real question that we in Wisconsin and folks all across North America have. As these CWD epidemics progress, what is the likely future of deer populations? And can we, if we wish, adjust harvest management in response to chronic wasting disease to potentially stabilize populations, just depending on what the goal is. But can we incorporate the CWD mortality into our harvest management decisions, which are a function of goals and such as well. In addition to that, what's the impact of chronic wasting disease on buck age structure and trophy opportunities? So I think what I didn't say is that prevalence tends to increase with age. So the older animals are more likely to have it than younger animals. That's mostly just a function. It could be somewhat behavioral, but it's probably mostly just exposure time. And then bucks, males, are more likely to have the disease than females. And again, that's likely behavioral, but we're not so sure. But at any rate, so an older buck is the most likely deer to have chronic wasting disease. And so those are the deer that are also most desirable by most hunters. And so as the disease spreads and increases in intensity and prevalence, how is that going to impact hunters? And so then once we know that, we can ponder what that might do to recruitment and retention of hunters. So to sort of get at these questions, we initiated this mouthful of a project that we call the Southwest Wisconsin Chronic Wasting Disease Deer and Predator Study. We initiated field work in January of 2017 with 4 years of deer capture, and it's taking place in northern Iowa County for the most part, which is again that endemic area, or what we consider to be the endemic area, just the highest prevalence area in the state. So a little more specifically, we want to, with our deer capture and our collaring I'll talk about, we want to quantify survival as a function of disease status, age, and sex. So when you take that survival or that mortality rate, or the flip side being obviously survival rate information, you combine that with recruitment, harvest age structure, and with CWD prevalence. When you combine that information in what we call an integrated population model, then you have a more comprehensive understanding of the deer population. And we can, the next step and the ultimate step is to quantify the relationship between the chronic wasting disease prevalence we observe and then the deer population change, or the growth potential. One more note about harvest age structure. So a subsample of the deer that are harvested, we, the DNR, will get an age estimate on those deer. That's demographic information. Age structure of the population provides information on the demographics of the population. So that's an important piece of the puzzle. So our field project is focusing on survival, and then we combine that in this modeling framework to get a full population level. And so what we want to do is to, again, like I said, quantify that relationship between chronic wasting disease and population change. And this is a hypothetical example or construction of that, which we want to quantify. So on the y-axis, you have population change, and then the x-axis, you have wasting disease prevalence. That black horizontal line is stability, so the population is neither growing nor increasing year on year. And then that red line is that hypothetical relationship. So the idea being in this relationship being that any chronic wasting disease prevalence reduces that population growth by some factor, and then at some point it's gonna enter that stability line, and then beyond that intersection point, then the disease on its own is causing population decline. And then on the left side of that intersection point, that's the space by which the population potential is being reduced, and that the closer you are to that, then the less leeway you have harvest management. So normally, you can think of that space between the black line and the red line as like, what's our, the old-fashioned term would be harvestable surplus. How many can we harvest year on year to have a stable population and still provide those sort of, the social benefits of hunting. So we want to put numbers to that. What is that prevalence where that intersects, and what's the shape of that curve, and have the numbers on that. So that's like the sort of the end goal that we're striving towards. And I should say, as I continue, we don't have this yet. So we just finished our deer capture. So I guess I should preface this when I started. I'm describing the study. We don't have results. I'm describing what we've been doing and what we are doing and why we're doing it, but not what we found, because we haven't found anything out yet, or not much yet, because we're still in the data collection phase of the project. Okay, so then the goals in terms of capture and data collection were to capture and fit 200 adult deer with GPS collars every winter, and then in the spring put 100 collars on newborn deer. So the goal was to do that every year for 4 years, starting in January of 2017. We'll get into the numbers later, but COVID was such that we couldn't do a fourth and final fawn capture season. So we ended up with, we did get 4 full adult capture field seasons in, but when COVID hit, that was after our winter adult capture was done, and then before fawn capture, so we couldn't do fawn capture. So we're with 3 years instead of 4 years. So when we catch these animals, we do a lot with them. We take tissue samples for genotyping. Again, I mentioned that it's called the PRP CWD genotype, and I mentioned that genetic susceptibility. So we'll get each deer's genotype with that. Pardon me. we'll take an antemortem CWD test. So then we'll have a test of their CWD status at capture, and then we also do body condition, body weight. We pull a bottom outer incisor. It's very small. We pull an incisor on those deer whose age we don't know. I mean, we can tell if it's a fawn or a yearling, but if it's older than that, then we'll pull a tooth, this little incisor, and then that will get aged just like tree rings. So we collect a lot of data at capture, and then we follow them through time and determine the fates of each individual. And then at postmortem, we'll do a postmortem investigation, and then another chronic disease test at death. Just a little bit about how we go about this. This is so the adult capture is all during the winter time. So we need to catch deer effectively. You need to bait them. They're not going to come to bait really as readily during the summer because there's lots of other food. So we use the time that's their natural time of deprivation for them to catch the deer, because that's when it's the easiest. That's the only time you can really do it. Even if you have a warm spell in the middle of winter, you're going to have a hard time catching deer. So we rely on snow and cold. Okay, so this is, we call a netted cage trap. It's just a metal frame with net on the sides. It's got a trap door in the back, and then it's got a tripwire 2/3 of the way from the door. We'll put shelled corn bait in the back, and then when the deer come in, they tripwire, the door shuts behind them. So with these, we'll use these on bigger properties. There might be 12 of these traps spaced out across the property along a back trail back deep in the property. And then mid-morning, our crew will drive the trap line and check the traps. So they'll come upon a trap, it's got a deer in it. Someone's going to have a snowmobile helmet or a hockey helmet. Usually, this is a bad photo because she doesn't have a helmet on for some reason. She's lucky she didn't get kicked in the face. But anyway, so they'll have the helmet on. They'll go in there and they'll kind of subdue the deer, get the deer in a manageable position like she does right there. And then she'll put a blindfold on the deer. That's it's a stocking cap at the end cut off of it. And you can see this other technician that's standing up. She's pulling a dose of anesthetizing drug. She's gonna go in there and give a dose of drug to the animal. They're just gonna be quiet for about 5 minutes, and then the animal is down, and then they can do the full workup. This is the other technique we use. It's called the drop net. So you see that flagpole on the left side. There's that net, that black net is suspended from that flagpole, and then the edges are also connected to T-posts and stuff at the outside. And then our crew over the years have invented and continually improved upon this really neat mechanism for dropping the net remotely. I won't go into describing it, but basically it's set up, and there's corn at the base of the pole. There's a crew member sitting somewhere, probably in a truck or a blind or a tree stand, some distance off. And then when the deer come to the net and to the bait under the net, and the deer that we want, they're going to drop the net on the deer. Then the deer is going to get tangled up, and then the rest of the crew is nearby, is going to drive up and drug the deer. Again, you wait, and then you disentangle the deer, and then you do the workup. This is just some of the kit that we use. The drug we use is really fantastic. It puts them down fairly quickly. They're usually very smooth. They're not moving around. They're not fighting when they're under, and they don't have complications. Typically, they don't have problems respirating or with their body temperature. That said, we continually monitor their breath, their heart rate, and their breath rate and their body temperature throughout the whole process. We're just periodically checking and recording it. But this drug combination that we use is really good. You can see the technician on the right-hand side, she's holding a collar. That collar is going to go in this buck, and the buck collars are elastic. There's that sheath there. Underneath that sheath is elastic, and it's got folds sewn into it. And so with bucks, obviously for one thing, they're going to grow with age, but especially with bucks during the breeding season, their neck will increase in size quite a bit. So we need a collar that's not too tight and not too loose. And so it'll kind of stretch and grow with the deer and then kind of retract to some degree afterwards. And so, but that's a GPS collar. So we'll put that on him, and I'll tell you about that GPS technology in a little bit. The females get a GPS collar too, but it's just not elastic. This is the antemortem test, or the source for that test. And so this is a rectal biopsy. So in this portion, just inside the rectum, in this tissue, has lymph follicles. And so we will take about a penny to a nickel-sized piece of tissue from that. It's like, in its natural state, it's like all folded up. And so you kind of use that green plastic, and that's a speculum, to kind of spread it out. You pull it up, you snip, snip. It's very fast. We have done this over 700 times, and we don't get complications. It bleeds some, but it heals extremely fast. And again, that tissue will be sent to Wisconsin Veterinary Diagnostic Lab for chronic wasting disease testing. Now, it's not a perfect test, or the tissue is not perfect tissue in that there's a false negative rate that's not huge, but it's important, something that we need to take care of. But it's very easily accessible tissue. In the past, they'll take tonsil biopsies or something, and that's much more invasive, much more difficult. This requires not very much training, and it's very quick and easy on the deer. That plastic speculum is commercially made for sheep, because sheep have another prion disease called scrapie, and so those being livestock, there's a market for those speculums, and we were able to buy those, and they work perfectly for deer. So that was pretty nice. So for fawn searching, it's a almost purely brute force effort. So we've taken on many hundreds of volunteers. So we're catching, end of May, the last 2 weeks of May, and the first week of June is that period when the fawns are being dropped. So we're trying to catch them right after they're being born. In fact, when they're a few days old, you can't actually catch them. You have to catch them right away when they're brand new. It's really hard to do that because they're hiding for good reason. That's what they do. That's what they do at that age, is they hide. It's really easy to walk past them. So what we do is we get giant crews of people, and we walk likely spots. And when we see a fawn curled up in the ground, we quickly grab it and do the workup. And it's a pretty quick workup. It's a few minutes. We do not do that rectal biopsy. They're too young to have it, and it doesn't make any sense. I should say the disease is not believed to be vertically transmitted, so it's not going to get chronic wasting disease in the womb. That seems to be the case, and so there wouldn't be any purpose in testing a baby anyway. And of course, you wouldn't want to do that thing you wouldn't want to do a biopsy on a newborn. Anyhow, no need to. So the workup is quick. It's just some body measurements. We're putting him in a laundry basket to get its body weight. Put a kid's sock with a hole in it as a blindfold, ear tag, and a radio collar, and send it on its way. We put these plastic ear tags in every single animal we catch. So they all have a unique number. So these numbers for these deer that are recruited into our study, this is their social security number. So all the information we collect, the capture information and the mortality information, the location information, it all goes into a database. There's tables and queries and such. And so every single deer has its own social security number, which is written down, obviously, of data sheets and stuff in the catch deer. And you can see the collar there. That's similar to that buck collar I spoke about. And you can see here the stitching in the folds really easily. And so again, this is elastic collar. It's got a bunch of folds that are sewn. That thread degrades. There's different thicknesses of thread or different numbers of threads for each fold. And so it's kind of over time, there's multiple folds. And so there's the first fold is going to drop after it gets such and such size, and it's just going to continually grow with the animal. Eventually, these collars will just rot away and fall off the deer if the deer lives that long. So it'll be on the deer for 1 to 18 months, sometimes 2 years, but usually not that long. and this is what they look like. For the most part, they're just lying there, and it's easy to grab them, and you set them down, they might trot off and lie down, or they might lie right back down. But so this has been also a really huge outreach opportunity. So we've gotten hundreds and hundreds of just volunteers to come out and do this, and it's always a great time for folks. So the technology that we use is pretty cool. The GPS collars that we put on our adult deer, they have an accelerometer inside of it. And when the collar goes motionless, it's going to give us a text message and an email with the collar number. And then it's going to put out a blast of locations at that site where it went motionless. And then that's presumably going to be the death location. So that makes it very easy for us to do our mortality investigations for the most part. Once in a while, it'll die on top of the collar, and then that alert will be delayed, and that hinders us. But usually we are able to get to them pretty quickly because of this technology. It's really handy. So now we're dealing with dead deer. So we're doing tons of, it's a mortality study. We're doing lots of mortality investigations, and so we get a mortality alert. We'll get permission on the property and go find the deer. The first thing they'll do, they've got a full checklist in a routine they go through, and they're doing sort of like a homicide investigation, right? So they're gonna look carefully at all the surrounding area for footprints, scats. What's the vegetation like? What kind of place is it in? Is there any sort of evidence that might give us some information about how it died? How close is it to a road, and things like that. Those two animals on the left, they were intact. So those animals are all going to be brought back, and then they're going to be taken to the University of Wisconsin the vet school, where there's a veterinary pathologist who does all our necropsies. So they do a very extremely thorough report on each deer that they get. That helps us in cause of death designations. When the deer are consumed, or at least mostly consumed, and then it's not possible for us to get in because the evidence is mostly ruined, and you can't bring in a deer, especially if it's decomposed. So then they'll do a field necropsy, and that is more or less elaborate depending on how much is left of the deer. So like in this deer on the right-hand side, they'll skin it out, look for any bite wounds, or they'll look at the organs. They'll look to see if they can see any obvious pneumonia and things like that. So like for instance, this deer on the right-hand side, well, we know it was eaten, but we know just from that whether it was predated or scavenged. So one thing they'll do is they'll skin it out, and they'll look, especially on the neck and the head, is there hemorrhaging? So if obviously if the animal is getting bitten while its heart is pumping, it's going to bleed. So there's going to be hemorrhaging around the tooth marks, and it's really easy to see those same kinds of tooth marks that happen after the deer is dead and not pumping blood, and that there's no associated hemorrhaging. So it's not a perfect way to go about it, but it's just they're sort of collecting as much evidence as we can to try to find out what happened. So they're also going to look for a chase scene, things like that might give us an indication of whether, for instance, like in this case, whether it was scavenged or predated. And then if it was predated, like was it really low on fat, some of those circumstances that might lead it to make it more vulnerable. So a lot of mortality investigations. So in terms of our field work results, so throughout the study, we collared with GPS collars 766 deer. We got 323 newborn deer, and that was again with 3 years rather than our 4 that we'd planned. And then these antemortem capture tests, we got 761, and then 79 of those came positive at capture. Now, like I said, there's that false negative rate, so we certainly missed some. Again, it could be a whole talk about this, like how do we deal with that detection probability issue, the other assays that we're working on that are an improvement on the current assay. So that number likely change once we make some improvements, especially with the assay. That's what we had at least to start. And of course, many deer that test negative at capture will test negative at death, and that could be either that they acquired the disease after we caught them, or it could be it was a false negative at capture. So this just shows one of our years of our of a survival analysis. So I showed you that survival analysis from that published paper at the top of my talk, and so this is us basically repeating it using one of our years of data. And so this is like a survival curve. So at the left-hand side at the top, that's one. That's they're all alive, and then as that line declines, that's representing mortality. And so you can find out the mortality to any date by finding the date you're interested in on the x-axis and running it up to where it intersects with the line, and then running it over to see what the survival probability is. The vertical line at September 16th, that vertical dashed line, that's the start of the archery season. And then if you move over to the right, the next dashed line, that little shaded vertical area, starts on November 17th. That's the traditional 9-day firearm season. And then the other one to the right of that is the December antler season. So you can see that's when most of the mortality occurs. So we can also see, this again.
So Dan?
Yeah.
Back to my earlier question about whether hunters are systematically harvesting more diseased animals. It seems like the slope of the line for the positive animals is steeper than for the negative animals. Is that right during hunting season?
It does seem like that, but I would say for this particular model, because this is a output of a particular model on the data, and it does definitely look like that, but I don't think in this iteration that it's allowed to differ, although it does appear to differ.
Okay.
I think in this, so think about a statistical model, this would be, there's a rate, the mortality, I mean, it's a pretty complicated survival model, but I think it's just like it's just the survival is lower by a factor based on whether it's positive or not. I believe that's the case for this model, although I agree with you, and that was the source of discussion. Like it does appear that that's the case, and it's going to take more data and more fancier models to really tease that out, though. But I appreciate you saying that, because it does appear to be the case.
Yep.
So, and then you see, so the lines then are, of course, those are the estimates, like the survival estimates at every point in time. Then the shaded areas are our 95% credible intervals. And of course, our credible intervals for our positive deer are much wider because there's much fewer of them. So this isn't anything final. This, again, is 1 year. It's just we want to produce this stuff early on in the project, just to sort of demonstrate to the public, hey, here's what we're doing, here's what we're after, and so people didn't have to wait 5-6 years because it's going to take that long for us to get the final answer from when we started. But just to kind of keep people engaged and stuff, we thought, all right, let's do this more basic analysis, with a lot more data. So the other thing is, and this is just irrespective of the age or sex of the deer. Well, that's really important, right? Because it's female survival that drives population change, not buck survival, not male survival. And they have different mortality, just irrespective of the disease, they have different mortality rates because they have higher, especially harvest mortality. There's much higher harvest mortality on bucks than there's does. And so we need to separate all of that out. Age and sex and status and potentially year, but we need to get all of our data together before we do that. But this just it's an example of, like, on a basic level what we're looking at. And so, I mean, we say, like, look, we just started the study and here's what we found. We've got a lot to do. We've got a lot of refinement, but even with just a tiny sliver of our data, we can already see, like, there's a big difference, because that's just from an effect size, it's a huge reduction in survival. It's not enough to say, okay, the population is going to do X, Y, or Z, because, again, we need to split it out by sex and age and integrate it with the other information. But it was sort of an eye-popping, not surprising necessarily, but it's kind of a big result, especially when you have so few data. I mean, this is just 1 year, and so in any 1 year, there's not a ton of negative or positive animals, and so anyway, that's where we are at this time. I know we got 10 minutes or a little bit more than 10 minutes left, and I can keep going as well afterwards. I just wanted to talk a little bit about some of the other stuff. I talked about the why we're doing this project, but there's a lot of other, there's a whole bunch of cool stuff that we can do with this data, things that we can learn especially because of these GPS collars. And this shows a single individual for a brief span of time in April of 2018. It's showing at this point in time, the collar was getting hourly locations. So you can see it. You can see the yellow are the locations accumulating, and the blue is just the dot as it moves through time. So there's a lot we can learn about deer, how deer relate to their environment. You can see like that there's a field to the right of the road on the very far right hand of the screen where it was feeding at night. You can see where it preferred to bed. You can see where it doesn't like to go. So there's a whole area of statistics based around analyzing location data and movement data. Another interesting thing here is really, I think, cool with this animal is, again, it was going to that one right field, and then it switched. Something happened in April where now all of a sudden that field on the bottom, it didn't go, it went to the far side of the field, not by the road. It didn't go to the field on the north side of the road. It didn't go east or west of the field it went to. It went to that field. It didn't go north of where it is bedding. It went to this one spot and this one field. Pretty interesting. It turned out that was, I think that was alfalfa that was starting to green up or something. So, and then it's interesting. So like he continues to bed where he had been bedding. His main food source is obviously this field. He didn't go bed in the woods right across from where his food was. He still went back north. Why is that? So there's all sorts of cool things. Like I said, there's a whole area of statistics developed around understanding this stuff. So you can classify the landscape according to the cover type, the type of forest, the type of field, the aspect and the slope and things like that, and analyze what sort of habitats it's selecting for and selecting against, how that changes through time, how that differs between types of individuals, seasonally, but then bucks and does and fall and spring and summer and winter, which is just interesting from a deer ecology standpoint. But this could have some implications for chronic wasting disease. So like once we start to put all these different individuals together and understand this, like where and when or what are the circumstances that might cause deer to aggregate or congregate together. So and what, like when is that happening? Where is that happening? What's facilitating that or not? This potential for deer that normally don't live together to be at the same place at the same time, or at the same place, maybe not at the same time, but just congregation, sharing space, because that's the mechanism for disease transmission. So there's a lot that we can learn there.
Dan, I got a question from one of the participants. Could you talk more about the risk to humans from consumption of, I presume the question is infected meat?
Yeah, so the current conventional wisdom is that it's not likely that people that eat infected meat will become infected, but that we don't. There's no certainty there. It's clearly people are eating infected deer all the time. The WHO, the World Health Organization, any sort of governmental agency will say, do not eat infected meat. We don't know. It's never been confirmed that humans can get it, but just to be on the safe side, because there's so little known about these diseases, that just the precautionary principles that you should not do that. So that's kind of where that lies. So what you're seeing here, this animation.
Sorry, Dan. I think I had another question from Uwe Pot.
Worked on PSE some 20 years ago in Switzerland, and it took a long time to clearly prove that it was transmitted from bovine to humans. Mm-hmm. And although there were human cases, of course, all over the place, but to make that causal relationship, so I don't know how many prion diseases we have in Wisconsin in humans, but maybe it doesn't necessarily mean that you can't show that it happened, that it doesn't happen.
Right, and I think most, and again, to me, this is like a prion. I'm a deer guy. This is a prion. This is a better question for the prion experts. And I'm just taking my understanding which is that there hasn't been confirmed, but it doesn't mean it can't happen, or it doesn't mean it hasn't happened, or even if they could magically have confirmed that it hasn't happened yet, doesn't mean that it couldn't happen in the future. So there's a ton of uncertainty around this, and again, is there even like differences in human susceptibility based on genetics? I mean, who knows? There's just a lot of complications there. I think the bottom line is, like, I think most people in the know, like, I don't think I don't know of any prion people who are like, yeah, who cares? Just go ahead and eat it. I don't think that's the case. I think most people are just like, be cautious about it. But we do know that there are many people, many people who are eating infected deer every year. And so in some sense, it's a great big experiment, unfortunately. And hopefully it remains to be the case that we don't confirm it, but you never know. So what this is showing here, back to this slide, is dispersal. So what a lot of animals do is juveniles will disperse, and with a lot of mammals and deer, it's mostly the males, but not exclusively the males. There's a percentage of those animals that will leave their natal range around their first birthday, and then they're going to disperse. They're going to go somewhere else. And so, and then some deer will also, those same deer will, or of that age class cohort, will also disperse in that following fall. So when they're about 18 months. Obviously, when you have deer moving, that's a potential for disease movement, right? So we want to understand that as well. And this plot I'm showing you, or this movement animation I'm showing you, is one of the more dramatic ones. Most dispersals are much less interesting and far. They're going to disperse 5 miles or less. But some are pretty far, and this is a far one. This was, I thought, it's just a fascinating one in that, in how it backtracked almost perfectly on its own dispersal path, and on how on earth they could perceive the landscape in that way is incredible to me. But it's interesting to think again. This deer was infected. This was a deer that got infected early in life, and of course, most deer, when they get infected, they're getting infected later in life. This deer would have had to be infected before it turned 1 year old. And that happens. It doesn't happen as often as older deer, but this deer happened to be infected. So understanding the role of dispersal in disease spread, because we know that the disease isn't spreading uniformly across the landscape. It tends to move more west than east. I'm talking about this area in Wisconsin, more west than east, more north than south. It grows fast. There's other research that we have that it grows, the prevalence increases faster in some areas than other areas. So it's not happening uniformly. So how does the landscape facilitate or hinder deer movement? Does it induce some directionality and dispersal that might influence the spread of the disease? With this really high quality information we have on lots of deer, we can model these dispersal paths and how the landscape enters into that. So a lot of really cool stuff we can get from this data to help us understand CWD. This is starting to wrap things up. So what we do a lot of outreach for this project. We've got a newsletter that we publish every couple of months, and people can subscribe to this newsletter. We've made early on in the project, we were making a fair amount of YouTube videos just to show people how, like I talked about, those traps and the handling and the capture and stuff, like how does it work? Well, we put together a bunch of YouTube videos to show people how we do it. We keep our up-to-date sort of collaring statistics and stuff. And so again, we put a high priority on communicating the happenings of this project. And again, I'm talking about volunteers. We did a back of the envelope calculation on our volunteer effort from our 2019 fawn capture season. So we had, in 3 weeks, we had 1,800 volunteer hours of volunteer hours. So that was huge. It's really good for, we're a government agency. We need public support, and this sort of thing is really helpful. And the landscape is almost all private land. So we're working on private land, private properties. We've done really well with that, having a lot of a really good positive response. And so we've had hundreds of landowners that we've cooperated with. So that's what I've got. So, but I'll take questions.
I'll clap for the crowd. Do people have questions? I've turned on chat for everybody. People could chat questions, or I guess I can modify the settings here. John, yeah, do you have something to say?
Just curious about your photo on the right-hand side there. Are those ticks on its back?
I think they're flies. It might be. I think they're flies.
Okay, I've allowed participants to share audio. If anybody wants to unmute themselves, they should be able to do that. We'll give people a few more seconds to ask questions, but so that's John again?
Yeah, I had a question. What is the typical variation in the position of a deer? I know deer generally don't migrate that far or anything, but are there some that go across the state, or do they pretty much stay within a 5 or 10 mile radius?
Yeah, so migration is really dependent on basically the landscape and the climate. So there is some migration in northern Wisconsin. There's more migration in like the northern UP, the Upper Peninsula of Michigan, northern Minnesota, in the western mountains. Deer populations are migratory. In the farmland, Wisconsin, they're not really migratory for the most part. I mean, deer may be vacating a property and going to where there's a good food source in the winter. That's not true migration. In terms of like moving across the state, I mean, that dispersal I showed you I think it was like 30 miles or so, and that's definitely on the longer side. I mean, there's a few, we're talking really rare dispersals where it may be upwards of 100 miles, but that's really uncommon. So even when they're dispersing, they're leaving their natal range, they're often not going. They're going a couple miles, 5 miles. I mean, they're just not going that far. They are, they tend to have home ranges that they stick to because they just become familiar with the area, and that's very advantageous for them to have familiarity with their landscape. They know where's the food, where's safety, and that sort of thing. So they're not just bebopping around the landscape all over the place. They are pretty tied to a place. Apart from that,
okay, thanks.
Yeah.
I have one more question here that somebody chatted in. Can you comment on deer densities over time? So, for instance, from maybe pre-European times, and deer densities in different counties. So, for instance, you say this one particular area. Is the deer population there higher than in other parts of the state, or is it kind of uniform over the state, or?
Yeah, it's definitely not uniform over the state. So first, and I can read her question. So thank you, Renee.
Okay.
So the pre-Columbian time, it's all guesswork, but white-tailed deer were common. I mean, this was before when the landscape, before the European influence of the landscape. This was white-tailed deer country as well. It would have varied tremendously across the landscape based on how much is prairie versus very deep dark forest versus the savanna that that's better for deer. I mean, they're not a creature of the wide open or the vast interior forests as much as they are on these more transition zones. And so how many deer were there now relative to pre-Columbian times? I don't think anybody knows for sure. I would be willing to bet that I would be surprised if we don't have many more. The reason being agricultural crops. I mean, we take so much of that. If you think about the energy on the landscape, like the consumable energy on the landscape, and you think about a forest and what percentage of that biomass is consumable by a deer. Now think about a cornfield or a soybean field, where soybean field, like almost all of it's consumable. So you're just how agriculture works is it makes way more food for the deer. At their level, it's highly digestible, and it's there every year. So with natural vegetation, there's this at least potential density dependent feedback. So the more deer you have, the more they browse, the less food is left for them in the future, and so they can't just keep increasing. But you've got a soybean field or a cornfield, it doesn't matter how many deer there are going to be, because they're going to plant a new field the next year. So there's more deer now than I wouldn't say ever, because maybe more deer now than ever, but maybe in the late 90s there were more deer, but certainly relative to pre-European times, there are way more deer now. And deer are not uniform in their densities in Wisconsin. In what we call the farmland, so most farmland tends to have, where there's any sort of reasonable component of agriculture, there's going to be more deer. The more heavily forested counties, so if you're familiar with like Iron County, for instance, Florence County, those deer densities are much lower because there's just much less agriculture. And there's been changes with the European settlement, especially there was the Great Cutover when they did a massive amount of logging in the 1800s, and then that grew up, and then as that during the certain stages of forest succession, that became really good deer habitat, and then deer populations boomed, and so kind of changes depending on what's going on with the landscape. But the deer densities in Iowa County are still pretty high. They're among that region is among the highest, but also Shawano, Apaca County, so it's a little bit west of you guys in Green Bay. That's also some of the highest deer densities, and so those are the counties where you have a really good mix of forest and agriculture. So the counties that are heavily agriculture, like southeast or south, the very southern extreme, where there may be a lot of deer in every bit of cover that there is, but since there's not that much cover, I mean, there is some need for cover. So the super high agricultural counties have fewer deer. The super forested counties have fewer deer, where in the middle is where you have the most deer. So kind of a long answer, but yeah, that's the situation.
Well, I think we shouldn't keep Dan much longer. It's past quitting time now. But I want to thank you, Dan, for taking the time to talk with us, and I want to thank everybody for tuning in.
Yeah, thank you. Thank you for having me, and appreciate your guys' attending and asking good questions.
Have a good weekend, everyone.
Yes, you too.